So good afternoon, everybody. As Carol said, I'm Mary Garvey, and I was a student here in AIT. I did my PhD here in AIT as well with Professor Neil Rowan. And my area, I've been a postdoc since January here in the college, and my area of research is to develop a pulse light approach as a novel solution for drinking water treatment in Ireland. And this project is funded by the Irish Environmental Protection Agency. So I just want to start off by giving a brief breakdown of my presentation, which will be short and sweet. First, I'll just give a little introduction into our area of interest and what we're trying to achieve in this group. I'll highlight some of the aims of the project and where we'll be going in the next couple of months. Then I'll just give a brief breakdown of the methods that I've used so far and rationale or why I've used these methods. And then I'll just give you a list of publications that we have to date relating to this research project. So UV light. UV light is light that falls in the electromagnetic spectrum between 100 and 400 nanometers. It's broken up into UVA, UVB and UVC. In relation to decontamination or the inactivation of microbes or parasites, whatever, we're looking at using UVC, which is around 264 nanometers. In the context of decontamination or in the lab, light is generated using UV lamps. They can be medium pressure or low pressure lamps, and they generally apply light to the treatment area in a continuous wave approach, where it's just continuously spread or shined onto the treatment area. So what we're looking to do then is to assess how UV light can be used best in water disinfection. So our system that we have in the lab at the moment is a PUV light system, and this works by storing energy in a capacitor, and it then releases the energy through the UV lamp in short duration pulses that are about 90 nanoseconds in duration. So what we're looking to do, or the question we want to answer is this PUV light system that we have, is it suitable for inactivating mainly Cryptosporidium, although we are looking at some other organisms in the water treatment plant level, and not only at lab scale, but also in water treatment plants. The parasite mainly that we're looking at is Cryptosporidium. I'm sure you've all heard of that somewhere. Cryptosporidium is a protozoan parasite. When it enters the host, it results in a condition or gastroenteritis known as cryptosporidiosis. Cryptosporidiosis has a 2 to 4 week incubation rate when inside the host, after which then there's usually a 2 to 4 week infection. In healthy patients or patients with a properly functioning immune system, symptoms can last for 2 to 4 weeks. However, in immunocompromised or individuals who are maybe AIDS or cancer patients, etc., the condition can last longer, and also it has been known to be life-threatening in certain circumstances. I don't think you can see it very well, but I have an image here that I've taken with our fluorescent microscope of Cryptosporidium that I'd stained with a fluorescent dye. Cryptosporidium is generally 4 to 7 micrometers in diameter, so it's quite small, and currently there's about 11 recognised species of Cryptosporidium. The two main ones that are associated with human infection are C. hominis, which is human specific, C. parvum, and C. parvum has been found to infect, I think, up to 150 different species of animal, but it's also very pathogenic in humans as well. Regarding outbreaks, or more specifically waterborne outbreaks, in Ireland the largest outbreak to date occurred in Galway in 2007. There was a water treatment plant had contamination problems with C. hominis, which is the human specific form. There was 90,000 residents affected in Galway itself. Globally, the largest outbreak to have occurred so far was in 1993 in Milwaukee, when Lake Michigan became contaminated, and that was C. parvum and C. hominis. So you can see that in relation to drinking water, Cryptosporidium can be quite problematic, and it's important that we try and address this in our drinking water supplies. So bearing that in mind, the main aim of this project was to see if we can use this pulsed UV light system that we have to rapidly and repeatedly and successfully inactivate Cryptosporidium in our drinking water supplies in Ireland, because there is a need to ensure a safe drinking water supply, and at present the drinking water supply in Ireland is regulated by the Urban Communities Regulation 2000, and there is no standard for Cryptosporidium in Ireland's drinking water regulations at present. So we have this system downstairs, and I have been using it, and I use it for my PhD studies here in the college. So the question that we're asking at the moment is, can this PUV system be adapted and implemented at wastewater and water treatment plant level? And if so, and if successful, will this system be more economical to use than the standard UV approach, which is the continuous wave lamps that are currently on the market in water treatment, for water treatment. So people or bodies who would be mainly interested in this project, our output of course will be the water treatment plants, the wastewater treatment plants, county councils, environmental health management technologists, and of course the Environmental Protection Agency. Also of course the consumers or the public who are very concerned about what they're consuming. So methods to date, up until from January till now have been using the bench scale PV system that I have downstairs in the lab. I've been studying a broad range of organisms to include gram-positive and gram-negative, and also Cryptosporidium parvum. I have been treating them with varying UV doses to determine the best optimal conditions for an activation before I moved on to Cryptosporidium parvum, because Cryptosporidium parvum is very difficult to work with in the lab. And this allowed me to get optimal activation conditions for the more culturable organisms before I moved on to Cryptosporidium. I'm also looking at using a range of bacillus spores as well, because at water treatment plant level, the treatment plant we're going to be using is in Tuam in Galway, and we're going to install the pulsed UV system once it's constructed into the treatment plant. So the idea is that I can spike samples and I can pulse and get back my treated organisms, and then can find out my viability. So because Cryptosporidium is a known pathogen and I don't want to be putting it into the water treatment plant even at the lab scale that we have there, I've been looking at using surrogate organisms. So I've been looking a lot in the last couple of months towards using non-pathogenic strains of bacillus species. So I had chosen Bacillus megaterium and Bacillus pumilus, which are non-pathogenic, and I have Bacillus cereus just as a comparison, which is a pathogenic organism. So I want to see if I can use these non-pathogenic strains as indicator organisms for Cryptosporidium. This is just one table that well, the more recent of the findings will say. This table shows the reduction obtained following the pulse treatment of various strains of bacillus at vegetative state. Sorry, so these are non-spores at 16.2 joules per pulse. So as you can see, with a low dose of approximately 1 microjoule per centimetre squared, we got about 3.4 log in activation of the non-pathogenic Bacillus megaterium and Bacillus pumilus, and there was a reduced rate of inactivation in Bacillus cereus. So initially it looked like Bacillus cereus was proven or the Bacillus cereus, the pathogenic strain, was going to be more resistant to UV treatment, but at higher doses of up to about 10.8 microjoules per centimetre squared, there was no difference, or no significant difference in the strain sensitivity to pulsed UV light. So at doses equal to or more than 10.8, we had no difference in the treatment of bacillus. That's in vegetative form now. So this table just highlights the spores, which are the main bacterial state that I'm interested in at the moment. So here you can see for bacillus spores, PUV treatment of Bacillus megaterium and pumilus, we got 1.49, 1.9 in activation with 2.1 joules or microjoules per centimetre squared of UV light, and we had only a 1 log in activation of C. parvum. But again, with increased UV dosages, we have up to 10.8 microjoules per centimetre squared. We have 5.3 in activation of Bacillus megaterium and a 5.6 in activation of Cryptosporidium parvum. And this is with my bench scale UV down in the lab, so you can see that there's very similar inactivation rates. Bacillus pumilus proved to be the most UV sensitive in spore form, but Bacillus cereus also proven to be most resistant. But again, if you keep going up in UV dose, this sensitivity is eradicated, or this resistance in Bacillus cereus is eradicated. And this also shows that, or suggests that Bacillus megaterium is showing signs of acting as a surrogate organism for Cryptosporidium parvum, at lab scale anyway. So my future work now will be to keep, I'm going to optimise it more for the bacillus spores and see if I can get, I have another couple of strains that I want to assess to see if I can get a very suitable organism for Cryptosporidium surrogate. And also I want to look at different methods for determining oocyte viability post UV treatment, because the method we're using at the moment is based on cell culture and infecting cell lines in the lab, which is very time consuming and it's quite labour intensive. So ideally for the lab, for the treatment plant in Tuam, we'd need something more rapid for viability assessment. This is just a couple of papers that myself and Neil have out since this project came up. I've submitted one in August on this bacillospore surrogate for Cryptosporidium work that I've been doing. That's submitted to the Journal of Parasitology. Earlier in the year, we got a paper published in Water Science and Technology, and it was looking at measuring the ATP levels of Cryptosporidium to see if that could prove a fast, rapid viability measurement of Cryptosporidium after UV treatment. And we've also got a paper out on the inactivation of other protozoan or bacterial endospores and Cryptosporidium in the Journal of Water Supply. And it's just a paper that was from my PhD, and also similar work using in vivo studies in mice and in vitro cell culture for the viability of Cryptosporidium. The project team, there's myself here in AIT, Professor Rowan is the project collaborator, dealing with Tuam wastewater treatment plant and installation of our UV machine there. We have engineers from NUIG, Dr. Clifford, Edmund O'Reilly, Martin Cormican and Maeve Duffy for ecotoxicology testing, which I'll also be doing probably next year on treated samples and downstream and upstream of water treatment plants. We have Dr. Andrew Fogarty also here in AIT. So just for acknowledgements, I want to acknowledge the Irish Environmental Protection Agency for sponsorship of the project, for Neil for, well, helping me out all through the years for the PhD and the project as well, and Dr. Hayes and Dr. Stocka for their advice and support, and all the staff in the Athlone Institute of Technology. That's it.